Welcome everyone to the latest installment of the SPARC seminar series. Today we have Dr. David Nickerson. He is part of the Data and Resource Center here at SPARC, and he will be speaking today about the visual exploration and dynamic interaction for SPARC. Very excited to have him here, show you what's new. And in fact, we just promoted the latest developments today, just a couple hours ago. So you have the hot off the press introduction to what you can do with SPARC. Before we do that, though, I'd just like to say that Dr. Nickerson is number 3 in our series, and you can see that we've got a great lineup of presentations from April until September. So I hope you'll join us for each and every one of them. The SPARC platform is a repository and an open neuroscience and systems physiology platform. It is NIH approved and a HEAL compliant repository. And as of today, we are powered by AWS. We've been accepted into the Open Data Sponsorship Program. And so that means AWS is covering and supporting the open data enterprise that we are seeking here on SPARC. So data sets are free, will be free to access going forward. So we're very, very excited about that. So what can you do with SPARC? We have a lot of interest in building not just a place for your data to exist, but also provide tools and a community around which, so that you can find data that you're interested in, meet people who have similar interests to you, and ask questions about applications or techniques that were applied. Additionally, because all the data that is contributed to the SPARC portal is fair, findable, accessible, interoperable, and reusable, it is at the ready for you to dive in more deeply, to utilize for your own purposes, to further it and extend your own hypotheses, or to replicate information that has been provided previously. So you can browse and get data. Dr. Nickerson will be talking about the anatomical maps in detail. We also have a computational and simulation platform. We'll be have a presentation on that a little later. And of course, we invite you to contribute your data, your models, and use what's available here on SPARC for furthering and accelerating science into techniques that bridge the body and the brain. You can do this in one of 2 ways, and actually probably even a few more ways if we get right down to it. But you can contribute your data sets individually, and when you do so, each data set will be professionally curated. It will receive a DOI, which you can reserve. So if you are publishing your data alongside a publication, a manuscript that is undergoing review, you can schedule those to come out on the very same day. This allows you to also build your body of work. So by linking your ORCID identifier to your data sets as you contribute them, we keep track and will update your ORCID profile with the assets and the DOIs for the work that you contribute to SPARC. It can also include code and simulation. So one of the things that we're really proud of here at SPARC is that it really is a domain-specific entity. We want to focus on the peripheral nervous system and what it controls, so organs and other systems. But we're not limiting it to a particular modality, technique, or species. So regardless of how big your data is or how many techniques it bridges, all of your data can stay together, and we would like to present it that way. We also can help you with proposals when you're applying for funding. So we can provide some great grant language assistance in order to help you demonstrate that you are planning to contribute to SPARC and its FAIR principles. It fulfills your DMS policy guidelines if you're submitting to the National Institutes of Health or other US federal entities. And as I said, it's an NIH approved data repository. So it's open and accessible for anybody to access and use. What do we have? We have over 600 unique contributors who have answered the call to contribute their data so far. And that includes over 11,000 samples, 4,000 subjects, bridging 54 tissues across 7 species using 26 methods. I'm sure this slide is outdated because we just did promote our portal just today. So I'm sure there is even more data sets that are available for you to utilize. That means across all of those samples and all of those subjects across those species and with those different methods, we've got 237 research data sets, 46 anatomical models, and 46 computational models, and 100% of them are available for reuse. So we really hope that you will come, enjoy, and use the portal and its contents to further your own research. Those data sets that are contributed, whether they be computational models, anatomical models, or research data sets, are accessed frequently from the portal. And this is a great way to see how your research is being utilized, not just the interpretation of your data, but the raw data itself and the protocols that underlie it. And so we've had over 12,000 downloads of the data sets and models to date. So with that, I'm going to hand it over to Dr. Nickerson. But before I do, I'm going to let you know that Dr. Naveen Jayaprakash is going to be speaking next time. And he'll be talking about the vagus nerve. So stay tuned. So with that, oh, this is Naveen. Whoops. And Andre, go ahead and take it away.
I need you to stop sharing so I can. Okay, thanks, Sue. Thanks for the great introduction. And despite the many Dr. Nickersons, most people across SPARC call me Andre. So apologies for any confusion in names. And yeah, as Sue mentioned, today I was just going to talk through essentially some of the things that help us enable the visual exploration and dynamic interaction with data and knowledge across the SPARC portal, largely focusing on what's currently available on the portal. And just to start with a disclaimer, as Sue mentioned, and perhaps probably poorly timed, as it turned out, the seminar was scheduled exactly when the portal promotion happens. So the latest version of the portal's just gone up. Some things may not quite be in sync, and some of the links possibly will look slightly different if you follow any of them. But we are always interested in feedback. We obviously do lots of testing to try and make sure things keep working, but it's good if you find issues to report them. And if you go to the portal and look in the footer, there's some very convenient links to provide site feedback that really help us improve things. So just before I start, we're really grateful to the support from NIH under the SPARC program for MapCore and the Data and Resource Center in general, which has really enabled all of this work to happen. So I'm part of MapCore. We're based at the Auckland Bioengineering Institute here in New Zealand, where the sun is just starting to come up through the rain. And so this is the current team. All of the work we're presenting today is done, by all the team. And also just a shout out to Jesse, who, after many years with us in the team, decided last year was a good time to go and spend a bit of time surfing his way around Southeast Asia. But he's also contributed a lot to the developments. We're also working, in the SPARC Data and Resource Center with the DATCH, MAP, SIM, and KCORE. And we particularly work with the KCORE team based in UCSD on a lot of the work we're doing. And a lot of the portal developments that we work on, particularly with Eric at DATCORE, and Dominic giving us lots of UI and UX knowledge and help. And we've also benefited a lot through this flat map change control board that we have with Janet Keys-Patrikoff and Marianne Martone, and until recently Kip Ludwig as well. So there's all the, I'm sure there's more people we should thank, but that's the ones. And I just want to shout out to the SPARC Codathon, which is on its way. And as you can see, there's various dates coming up, lots of information on the website. And if you're interested in winning some nice cash prizes, I highly recommend getting involved. It's a good way to spend a weekend. And then, as Sue mentioned, and as announced today, possibly in a newsletter that's just gone out, the SPARC Portal is now powered through this AWS Open Data platform. And if you want to know more about it, you can, again, on the footer of the SPARC Portal, there's the nice logo, which will take you through to the page that tells you all about that initiative and the way AWS are helping to power the SPARC Portal. But I just wanted to start really talking a bit more about FAIR. I think we use it a lot. Hopefully most people know what it means. But just to sort of lead through a specific example, just to get us all on the same page. So if findable, you know, a lot of this relies on things having persistent IDs and especially globally unique, so you can always find stuff. So through the SPARC Portal, you know, data sets as they're published, they get a DOI, which gives you a nice, you know, persistent, globally unique identifier to refer to your data. So in this particular example, it's some stomach vasculature from a rat. And along with that, the data set has a lot of metadata, and that provides this very rich description. And if you go to it on the portal, you can sort of browse your way through a lot of that data and metadata. And interoperable, so this is really the idea that the data and metadata should, you know, be in these sort of standard formats, be accessible in these sort of things that are relevant to the domain. So in this example, you know, another DOI, give me another data set. In this case, I'm tracing off some innovation data, again, in the rat stomach. And so, you know, in this case, that's sort of segmentation tracing data is available in the standard neuromorphological file format, which we typically call the MBF XML format, because that's the tools we often use. So that sort of using these standards really helps to make the data and metadata interoperable. Accessible, you know, so essentially things should be retrievable using standardized communication protocols. And so if you're into protocols and look at the SPARC Portal, you know, clearly we're using HTTPS as that protocol. But importantly, this is, you know, the data and knowledge and metadata is available both by humans and by machines. So as an example, there's the SPARC Python client, which will let you sort of interact programmatically with the data, which is quite useful. And then reusable, you know, a lot of this relies on provenance. So, you know, having that sort of knowledge about where these things come from. And so just carrying this example through in the rat stomach, you know, there's a data set, which is a scaffold map, which is basically integrating data from three different sources. So 3 different data sets. So reusing that data to produce the scaffold map you can see here. But then, of course, this is available as a data set, which in itself is also reusable, and you can do different things with it. And using, you know, tools like the scaffold mapping tools, you can grab that scaffold map and do some things with it that might be interesting, such as generating a computational model for future use. So with that, you know, sort of background of FAIR data, sort of going into the SPARC Portal, and that is sort of really what lets us then have these interfaces on the portal for doing these sorts of things, to make them available to help sort of, you know, enhance the findability and accessibility. these things and to make the artifacts themselves available for reuse. And so, with that, that's, what we need to power these kind of interfaces that lets us browse through these things visually, interactively, connect all these things together because they're using standards, they're using common terminologies and all these things. It lets us sort of really connect these things together. And so that, shows in something like a whole body scaffold that we have, you can have very detailed anatomical structures and link those to specific data or between each other. And then present these things in these sort of interfaces that let you see the data and interact with it in hopefully intuitive ways, but always looking for feedback if people see of other ways we should show data. And so having that sort of knowledge in there, it then helps us to be able to essentially find stuff, right? So you're looking for things. So again, the common terminologies help in the way that you can search across the maps, visually, interactively. We can suggest things that you might be looking for. And so in this case, you start in the search box here, typing left, and you can sort of see all the things that have left start getting suggested as what you might be looking for. But then different things are different. So we use common terminologies across all of these things, but different data sets, different maps have different features. And so you don't always have, exactly the same thing everywhere. And that's really where the ontologies start to come in. We can start to know, that while in a particular map, you might have a transverse colon, region, you don't have a colon as a whole, and in other maps you may just have a colon, but the ontologies tell us that these are the sort of the same things, or at least parts of the same thing, which then becomes very important when you want to start trying to sort of render or connect, data and knowledge to the maps. But having this, it also then lets us use these as a way to visually interact and explore all of the data in the SPARC portal. So if we take the rat, if you just look at it at the whole scale, you get these sort of markers that telling you where the data is. And this is sort of quite a zoomed out view. So you're just getting one marker to say, at least when I took the screenshot at some point, there are 258 data sets available. And you can have different ways to then try and explore all that data. So things like facets, where you can specifically choose different anatomical things you might be interested in. And on the portal, you'll see there's other sorts of facets that you can use to interact and filter what you might be interested in. But you can also just visually zoom around and explore these maps. And as you zoom in, sort of those data set markers uncluster, and you start to see the distribution of data and the different anatomical features across the map. And then when you choose something, so in this case, the searching for kidney, there's one data set. Again, at this point in time, could be more now. And you can see things, do these sort of things visually. Where your search results here show up in the map showing where the data is. In this case, there's one data set that has kidney and liver, so you're getting these two markers on the different anatomical features. And you can do things like interactively highlighting things as you mouse over, just to really help emphasize the sort of location and distribution of these data. And so then if you do sort of other sort of searching, in this case, it's just searching on Cheng, which I know to be someone's name who happens to have a lot of data. And so again, you see, you can get sort of the highlights of all the data sets in that list of results. You can see through the data set markers, the distribution, the different anatomical features covered by those 58 data sets. And you can start to sort of explore that data and see, what is the data, what might be interesting, what is it you want to reuse in your own work. And using this sort of interface, you can do that all in place. So through the sidebar here with the search results, it's giving you these cards that are essentially summarizing a given data set. And so using that, you can use this interface to then page through very quickly some of the information in there. So for this particular data set, you can see it's got 6 images and 4 segmentations available for interaction on the portal. That may not be the entire data set, just a subset that's been enabled to sort of really use it on the portal. And you, just without leaving the maps or losing your context, you can quickly page through and see those things in place to really get an idea of what the data is and how it might be useful to you. And see other data sets are different segmentations, and again, just sort of quickly paging through. In this case, the data has been mapped to a scaffold, so you can see, that very quickly, and you can then sort of say, okay, which is the data that's of interest. You can load it up in the actual viewers and interact with it, get a better feel for it. The portal has these interfaces to let you build up these sort of visualizations. And then, if you find something that's particularly useful or relevant, maybe you want to make sure you don't lose it or share it with someone else, you can then grab a permalink using the little permalink button, which will give you a sort of a URL that you can then use to come back to exactly the same view, the same state, same set of selections in the portal. And you can share that with people. You could use it in your publications to get the same sort of thing. But it's not just the data sets, right? There's a whole bunch of knowledge behind all this, particularly connectivity knowledge, which is also very important, and we want that to be fair, so things are findable, accessible, interoperable, and reusable. And that's really where the sort of SCAN connectivity knowledge base comes in, which tells us, all of the knowledge that powers each of these little lines that you see. And how these things are connected together, as well as starting to see how, some of the data or other sort of literature might be relevant to each one. So just to sort of, look at how do we build these maps based on that sort of fair description of the connectivity. So we start with the maps are basically hand-drawn anatomical diagrams. They sit on GitHub. They look very similar to what you see on the portal. They have these sort of manually drawn lines which can help route connectivity through some of these complex anatomical structures in a way that might be nicer to see than if you just draw straight lines connecting point to point. So we can use these sort of manually drawn lines with the annotations of these anatomical features to then render that connectivity. Then from SCAN, the SPARC connectivity knowledge base, which you can find more information in this paper, we can pull out that connectivity information and using our software, which is also all open source and available for anyone to use, we can build these maps. Those maps, when they're built, they get put on a server, typically sitting in databases, with this sort of map tiles for the map interfaces themselves, along with all the metadata. And then the viewer that you see on the portal, which then lets you see these maps and interact with them. But in order to make sure you can actually, keep track of these things and use them and reuse them with confidence in future, keeping track of the provenance for all of this is quite important. And has been quite an issue over the years as things have evolved. So we try really hard to keep track of all of this information about where these things come from. So for example, SCAN, publishes releases, and that information is sort of embedded in a SCAN release, so you know what it is, and you can always sort of unambiguously then go back to know exactly what set of knowledge was used to produce any given map. Those sources on GitHub are essentially SVG files with a lot of annotations captured in JSON files. And so, using the versioning on GitHub, you can know, and sort of things like releases to know exactly what version of everything was used. Then when you make an actual flat map, we're using UUIDs as a way to track, very specific version of a given map. And so with that identifier, that tells you everything you need to know about what went into that map. So that on the portal, any given map at any given version, you get that UUID, and that will then tell you exactly what version of scan was used when the map was generated, all the different sources that went into it, the different versions of the software that was used and things like that. So you can try and know, if in future you find bugs or something in your software, you can find the versions of the maps that were built with that software to know what particular issues they might have, for example. So with that, we're pulling in all of this connectivity knowledge, which then lets you see, the sort of information about given very specific connections that are present in a map. And sorry, so when you're looking at a given connection, that information again appears on the sidebar. Here now, instead of data sets, you're looking at the connectivity knowledge. You get a card that tells you about it. You can see the description of what that connection is in sort of a list view. You can look at the sort of graph view. These sort of connections are essentially connected graphs that let you see that knowledge. And what you can do in the latest version of the portal that just went live is you can see that connection based on either the knowledge in scan or the knowledge that's in the current map that you're looking at. So as I said, some things are not quite the same in different things, but the ontologies let us know that. So this particular connection scan says, this is what the connection is. And what you may notice is that the one difference in this particular rendering of the connectivity is there's this bit here that goes through the ascending aorta. In this particular map, we don't have a feature called the ascending aorta, but because of the semantics and the ontologies behind this, we know that that's just the part of the aorta, which we do have, so we can essentially route that connection through the aorta instead. So that's until this release of the portal has been sort of silently happening in the background, and you wouldn't necessarily know. But now we're sort of making that explicit so that you can see these things on the portal and see these differences, which I think really helps to then understand how different connectivity knowledge from scan is actually rendered on a given map. And that's quite important because now there's becoming more tools that let you interact with that knowledge from scan. It's quite a few more tools are coming out now, which you can see here. And so, the maps are sort of the way that we've been showing that connectivity knowledge on the portal. And what Keiko have been working on is other ways to sort of interact with that knowledge, both in terms of curation and creating that knowledge through a tool called Composer, which lets you sort of really review and correct and then add new knowledge. But there's a new tool called Scanner, which lets you explore all of the connectivity. And I'll show a bit about this new natural language interface for Scan as well. But this is quite important because as soon as you have multiple tools trying to show the same knowledge, you find what's different between these different tools. So it's very important to know, which version of the tool, which version of the knowledge is being used in any given tool, which then helps to know that, okay, in this map it's rendered this way because it came from a different version of Scan, for example. And so these tools will be available through the SPARC apps. You can already now see the scanner there, so you can get to that through the SPARC apps page on the portal. And as sort of more of these tools reach sort of a level of maturity where it'd be useful to share them, they'll be added to this catalog of tools. But if you're interested, please reach out and we can point you to the sort of current test version of these things. So then the scan natural language interface, is hot off the press. It's very recent developments that the team in Keiko have been working on, but it lets you do, express queries in this sort of natural language interface. So you could do something like trace the autonomic pathways to the ovary. That will give you a set of results like this, which is often more useful to view as a graph. And it's really starting to now let you sort of more interactively explore the knowledge that's contained in the scan, which is quite useful. And so you can start to see these things, using the sort of all the emerging AI technologies that are helping you explore this kind of knowledge. Because it's very much trained on scan, it only includes connectivity knowledge from scan. So it's not, pulling in any sort of hallucinogenic extra stuff from the internet. So it's very reliable and sort of give you much better and cleaner results than if you were to just use general sort of LLM tools for this type of thing. And it, is really starting to open up these possibilities of different things you can do. And we're starting to think how we can integrate some of this on top of the maps to help, explore and discover connectivity knowledge. So as we look to have more control over what we can do through the connectivity knowledge, we need better ways to try and surface that information on the portal. And if you ever tried to use the maps in the past, you might have been frustrated that you often can only ever see one thing, one connection. So as of a few hours ago, you can now see more than one connection. So we're trying to make it possible to enable these things. And this is the sort of first step towards that idea of being able to show a whole bunch of connections based on some criteria, such as a result from that scan in our NLA interface. And so when you do this, what you can see is that different maps have different things, obviously. So looking at the human female, you can see that it's showing 237 different connections. If you look at the male, there's only 195. And because the rat is a everything animal, there's the sort of most that you can see is 292 different connections. But using this interface, it starts to let you see, more than just one connection at a time. And gradually over the coming month or so, we'll start to be seeing a lot more features coming out that make use of this in ways that help you explore the connectivity and data on the portal. So, a list of search results is good, but you need to use it. And so as you go through and do things like mouse over the given results, it highlights that particular connection on the map. You can expand it. You can see all that information about what the given connection is. There's little buttons that help you, locate it on the map if you've panned, your map off to the side or something, and you want to see back where it is. We're also starting to add these sort of convenient sort of copy and paste buttons to everything. So for example, if you were to click that, it will essentially copy all of this knowledge to your clipboard. So you could paste it into somewhere else. And importantly, I think, when you do that, it includes all the identifiers. So if you were keeping this in your notebook or something, it's not only just some text, but it's the actual identifiers that underlie all of this, which then really help you, find further knowledge, or if you wanted to reuse this. And so then, so you get all the information, list, graph views, whether you're looking at scan or the knowledge from the map, for example. And then, each of these terms in the list, you can now search for them. And so this particular term, pelvic sphincter nerve, you could launch a search right there to say, show me all the other connections in the map that happen to involve this anatomical feature. And so then you'd get a list of results. And again, here it's sort of showing visually all the connections that have, some either going through or terminating or originating through this particular anatomical feature. At the moment, it's very much just an anatomical feature, but we're also working on ways to actually use the knowledge that in this case, it's a natural nerve. And so we can do more with it than just treat it as a feature. And so, along with the connection knowledge itself, you can link to the data, find data that's relevant to each of these things, but also have all this information about, where the knowledge came from to establish this connection. And that's sort of where Composer comes in and helps sort of go through the literature, adding these either literature citations or hopefully data set citations as well, where there might be evidence for a given connection. And that all works because we have nice, unique DOIs or ORCID IDs for people that help, link unambiguously these things together. And so another thing you can do with this show related connectivities button, if you hit that, it'll show, sort of the other connections on the map that happen to use the same citation, the same reference, which is quite handy and quite convenient. So those are the sort of new features that have just gone up. Hopefully they work as well for you as they do in our testing. But really starting to sort of show more the entirety of the connectivity knowledge base rather than just looking at single connections one at a time, which we think is a big improvement, but we'd always love feedback. So another feature that's just gone up, we've had the ability, for people who have been given access to, essentially draw annotations on the maps. So you could interactively draw things on the map, potentially to show where there's errors, to highlight where there's missing knowledge, things like that. But we've added a new feature now where essentially anyone can do that. So on the maps, there's this sort of settings button down here, and if you go there and go into the annotation mode, you get a new toolbar which lets you start to draw on the maps. So in this case, I'm going to, I want to draw a line. Maybe I've spotted somewhere where my favorite connection's missing and I want to contribute it, or I just want to highlight something to show someone else. So in that tool, you can start to select points. Those points may be, anywhere, but if they're on an existing feature, sort of when you are on them, it sort of highlights the existing connectivity. So maybe it just helps you see, oh, actually the thing I'm looking for is there, but maybe it's not. And then you can sort of draw, it's perhaps a bit hard to see, the dotted yellow line that I'm drawing here, where I've sort of saying, okay, I'm drawing from here to this part of the vena cava. And again, it's sort of highlighting the existing connectivity as you go. And then completely made up example, this should be connecting through to this internal carotid nerve plexus, which again, lights up a bunch more connectivity, perhaps showing something that already exists that is there. But I'm determined on saying, no, I want to make something new. So then I can confirm that it's telling me what those features are that I want to connect. Then you get information about that. Generally, you don't care about these things. It's sort of more for people who are editing these maps directly. I can add information. I might suggest evidence through those different means, like a DOI or PubMed ID or ORCID ID or whatever, and add some comment. This connection should exist. So preferably, it would be a natural thing to describe what you're trying to do. All right, and then you submit that once it's there. So then exists and it's created on the map that you're looking at. And now, because you're not logged into the portal, it's got no knowledge of who you are or anything like that, so it can't associate, your information with it. But what you can do is, if you've made annotations in this way, when you create permalinks using that button, you get an option to say, do you want to include those annotations when you make the permalink? And if you do that, because we're always worried about sustainability and potentially, this is always, storing knowledge that has to exist somewhere. We don't want it to build up too much. So for now, we're saying, if you do that, it'll be available for 30 days. Potentially, if you want something longer, reach out to us or just go there and create a new one. And so having done that, you get again a permalink, and assuming things work as well as they did when I tested it, if you go to this link, you should then see that same line that I drew along with my comment. So those are the sort of features that have just rolled out, hot off the press, as Sue mentioned. And we think it's starting to really sort of enable the exploration of that connectivity, let us connecting to the data in sort of more interactive and visual ways. And if you're interested in sort of an example of how this sort of fair data is used in the more sort of computational and simulation pipelines, there's a webinar in a few weeks from the SimCore team, which will sort of really go into depth in that. And from the list Sue showed, I see there's now a seminar that it looks like Marianne will be doing it also in a few weeks, that will probably go into more detail around the scan. So I'd recommend definitely going to those if you're interested in more details. But with that, I think it's a very quick sort of summary of where things are at. One more plug for the Codathon. If you're interested, sign up. It's a lot of fun. And thank you for your attention. And that's pretty much what I wanted to show.
Thanks, Andre. One thing I'd like to add about the Codathon is that it's virtual. You do not need to travel anywhere beyond your living room to participate. So it's the first weekend in August, and we will build teams based on who signs up when the team sign up opens. And it's a weekend of playing with the tools that we've created, using them in different ways, suggesting other tools to be generated, and having an opportunity to really get to know the types of data and how you can take data sets from very diverse labs or systems and put them together because of the structure of the SPARC data set, as well as all the other open source and well-documented tools that we have. There's a quick survey that's popped up on your screen. If you wouldn't mind filling in those questions, we'd really appreciate it. But while you're doing that, I'm going to ask Andre a question that has come in. If you have any questions for Andre, go ahead and type them into the question window, and I'll be sure to ask them. So Andre, a question has come in. It says, the example of innervation of the ovary is excellent. So to be clear, this is based only on data in SPARC. Is that correct? It does not include published studies in the literature that have not been uploaded.
So it does include knowledge from published literature, but only the published literature that's been curated and contributed into SCAN, the connectivity knowledge base. So in those ones, the actual data has not been contributed, but essentially the citation from the literature and the knowledge that's been extracted from that is what's gone into SCAN. hopefully over time we're going to see more and more where it's the actual Spark, or data sets themselves that have been shared and made available that would be feeding into that. And the mechanisms are all in place to link to those and include that information. So I think the answer is the good old yeah, nah. But the literature that, knowledge from the literature that's been contributed, but not always the original data.
Right. And so I do know that we go to the primary literature when we're researching connectivity information to include into SCAN. But that does not mean that we're aware of every paper for certain for every connection in the peripheral nervous system. So we really invite the entire community to make this a resource that reflects their expertise and share with us these evidence containing manuscripts so that we can augment the knowledge that's held in SCAN. The person who sent in this question was thinking about this on the basis of like how a reviewer of a grant might review the information. So I think that's really pretty fabulous.
Yeah, and I think, part of this making that knowledge of what's gone into a given set of connectivity knowledge is very important, and having those sort of copy. I mean, it's a very trivial thing, right? Copy paste, but to make it really easy for people to copy that knowledge, put it somewhere. So, I showed an example of copying the entire set of knowledge, but you can actually, one by one go through the references and copy them in different ways, to include in things like your own bibliography, as well as searching.
I really like that feature of being able to select individual anatomies and say, show me other populations, show me other things that use that same region of the body. I think that that's really pretty fascinating. And then combining that with the ability to, for any given connection, when you look at the references, to be able to say, okay, well, what other information was sourced from that same manuscript is also really pretty fabulous. The information that is held in SCAN is not only sourced through published papers, as you indicated, it can come from data sets, and it can also come directly from experts in the field. And I know that Dr. Jeff Ardell was instrumental in providing some great representation of the cardiac circuitry. And it is something that it was a pleasure to be able to work with him on all of that integrative circuitry. So getting beyond just a small look to a greater view of the entire circuit. So I think that was something that really sets this knowledge base apart, and the fact that you can reflect that, expose it, and allow us all to interact with it different ways, I think is really, really, really cool. I do have a question about, so you've got multiple species on with multiple maps. And can you explain what the multi-flat map is and how we would see specific, like how do we navigate that?
Okay. let's see, where's it gone? Yeah, I'll just,
Did I put you on the spot? I didn't mean to.
no, I'm just gonna risk a live demo. See if I can break things. So, so we're on the portal, looking at the maps. So essentially, the multi-flat maps, because none of them are really a single species, they all contain a lot of different knowledge. And so for a given map, there are options to see, the different connectivity, where scan specifically says, here's evidence, that this has been observed in a given species. So it's important to know that this is just saying that someone's seen that evidence and added it. So the knowledge base is not to say that it doesn't exist or anything. It's just saying that this bit of knowledge has been added. And so in the interface, you can go through and you can see, okay, well, here's a set of connections that have evidence in this. If we basically, you can turn off everything and show, say, just show me the things where there's specific evidence that's been contributed that says this connection exists in human. And so you can see that this is that subset of all of it where there is human knowledge. There's, a lot in the mammalia, although the sort of balance is gradually shifting as more and more knowledge is getting added, and especially more and more knowledge of species. And so they're all the same. Connectivity gets rendered where possible. And so you see those differences that I showed in the sort of number of connections that are available on a given map, because the connections only get drawn if the features exist on the map, which also gives you that sort of difference between the map connectivity versus scan connectivity. So the rat typically has everything because that's just got everything, both sexes, and all features essentially. So when you're looking at that connectivity explorer, that's why you're seeing so much, because everything's drawn on the rat compared to the human. Is that what you were asking?
Yeah, absolutely. I think that that's fabulous. So you can see pretty simply based on what I guess backdrop of the species, what information is available and known to exist in different species, but also get a better view of what else is present.
Yeah.
That's really cool.
Yeah, and in terms of the data sets, essentially all data sets are available on all maps, but you can also then, use the filters and facets to say, okay, only show me data sets from mouse. What I probably want is rat.
And those data sets aren't utilized in scan, right?
No, they may be. So yeah, so the data set explorer is showing you all of the entirety of data sets published to the SPARC portal. Some of which may get used for different things, and the connectivity explorer is really then that sort of scan based knowledge.
Awesome.
And the sort of link between them, where you can say, okay, show me if any data exists. And in that case, where you're exploring or searching, you're just finding data that's related to these anatomical features. It's not specifically saying this is evidence for this connection. If it was, it would be more in the reference
Gotcha.
list.
That's perfect. Okay, awesome. Well, I think we've run right up to the deadline here. We're at the top of the hour. I want to thank you again Andre, for making yourself available so early in the morning and giving us a tour of the fresh off the press Portal release. Not only that, but I sweet talked you into doing a demo live of the freshly released Portal. So if that's not exciting, I don't know what is. So, but I am grateful for you for taking your time. I hope everybody is excited about the exploration possibilities that you've just laid out. And hopefully everyone will join us next time for our latest installment in the SPARC seminar series, where we'll hear about the human vagus nerve.